So kind of at one level, we want to understand algorithmically what's happening in motion detection, right? So how do you describe the transformation from visual inputs to an eventual motion signal? And this would be the kind of like mathematical equation you could write down on a whiteboard. At sort of one level down from that, we want to understand what the mechanisms are that implement these mathematical operations, right? So how do the cellular biophysics affect this? How does synaptic connectivity, synaptic biophysics, and so forth? So that's basically how do you do this math? And then at sort of a higher level, we're interested in understanding why evolution has converged upon this particular solution. And to do that, we want to understand the ethology of this circuit, how the circuit relates to the animal's behavior, and also how it relates in particular to natural inputs. So this is something we think about a lot in how natural scenes relate to the algorithmic structure of motion detection. And I think one of the things that's sort of exciting about this is that in these two levels of understanding the circuit really seem broadly applicable, right? So the mechanisms that I'm not going to talk about today are going to be sort of specific to a particular animal, but the algorithm and its relationship to the purpose of the circuit for the animal seem like these are sort of principles that are going to apply broadly, and that's so far our experience. So motion detection is really an inference problem. So you have an eye that looks at the world, and it has an image. It's an image-forming eye. And so you have a bunch of photoreceptors that are signaling to the brain what intensity they're seeing at each point in time. And the problem for the brain is to take this suite of inputs and figure out how to manipulate this suite of inputs in order to extract a useful signal that can guide its behavior. Right? So what I want to do is just look in very broad terms at how brains tend to do this. So I'm going to show you sort of responses of cells, of three different broad classes of cells to two different types of stimuli. So this stimulus is preferred direction stimulus, so we'll see why it's preferred in a moment. But this would be like a light edge moving to the right across the screen. And then this other stimulus would be, for instance, a light edge moving to the left across the screen. So the first thing that most visual circuits do is they process these stimuli with sort of non-direction selective operations. And so for instance, a single neuron here would respond to the preferred direction stimulus in the same way as to the null direction stimulus, right? And this means that it is not direction selective. So sort of deeper into the brain, you'll get to first order direction selective neurons. So these are the earliest direction selective signals. And what does it mean to be direction selective? It means that these neurons respond to motion when it's in one direction, but not when it's in the opposite direction, right? So here you can see this particular neuron would respond to, this would be its preferred direction, and this would be its null direction. And I guess one of the things that's interesting about these sorts of responses is that the null direction basically just has no response, right? So it's actually kind of difficult in some cases to tell whether there was anything there at all, right? And that's different from one sort of layer deeper in the network, where you sort of see these second order direction selective neurons. And what's very typical for these second order direction selective neurons is that they will respond with one polarity to motion in one direction and the opposite polarity to motion in the other direction. And this phenomenon is called opponency. And it's going to be like basically the primary thing that we're going to talk about today. And one of the reasons that sort of it's interesting, we'll see in a moment, but it can be generated in kind of an easy way from these first order neurons, right? One can think about it very simply as just having like one neuron that is tuned to one direction, another neuron that's tuned to the other direction. You have one of them excite this neuron, one of them inhibit, and you sort of get this phenomenology out. And so this organization is present in many animals, and including primates like us, and including more adorable animals like fruit flies. And these two species had a last common ancestor around 500 million years ago. And so it's very unlikely that this is an accident, that they have this sort of similar processing. And this sort of structure seems to have been converged upon by these two different systems. And we're very interested in understanding sort of how this all works. And so the big questions we want to ask are, how does direction selectivity arise in these circuits to begin with? In other words, how do these first order direction selective neurons become direction selective? And then we're going to ask where direction opponency first emerges. So what I told you about was that it looked like it showed up in these second order neurons, and we'll see whether that's really the case. And then last, we want to understand what computational benefits opponency might bring to circuitry and to coding of visual scenes. So we could study this in a lot of places, and the human brain is one, but we're going to do this in Drosophila, which is roughly to scale here. And there are a lot of advantages to going after this type of question and these types of questions in Drosophila. So one of them is that the brain is just small, right? So that means it doesn't have many neurons, and that it's accessible, right? So you can peer into the center of the fly's brain without removing a bunch of it first. Second, it has morphologically and genetically identifiable neurons. In many cases, we actually know the ground truth connectivity between these neurons. So that makes the sort of understanding these circuits a little bit easier. There's sort of robust behavior. So flies have behavioral responses to directional motion that are easy to measure. And then because it's in Drosophila, there's a lot of genetic tools that you can apply to trying to manipulate and measure from the circuit. And then last, there's a pretty long history in insect research and motion vision in particular of applying computational models to try to understand what's going on in the circuit and how it relates to behavior. So I'm gonna start just with a quick summary of what's gonna come during the rest of the talk. I'm first gonna replace this sort of generic diagram of the visual processing stages with the fly's specific circuits. We'll talk a little bit more about these in a moment. So the first thing we found is we actually discovered that opponency appears to emerge in the first set of direction selective neurons in the fly. So the first part of the talk is going to be going through our evidence for that. And it turns out that this fact actually really strongly constrains models for direction selectivity. So there's a long history of trying to generate mathematical models that would describe cellular function at this first step of direction selectivity. And this directional opponency result that we have puts very strong constraints on what possible models could describe that. And then last, I'll briefly show some simulations that we've done to try to ask what computational advantage there is to putting that sort of opponency in the earliest direction selective neurons. Okay, so the first thing I want to do is kind of dispense with the classical definition of opponency, because I think it can be misleading. And it can be misleading for the following reasons. So if you have a neuron and you need it, in order to detect opponency, you need to be able to detect upward deviations in activity and downward deviations in activity, then it means that you can miss inhibition if the basal rate of activity for that neuron is low. So for instance, if you take this neuron and instead of having it at this sort of mid-range level activity, you lower its activity to near some 0 point. And this 0 point could be like 0 calcium concentration, it could be 0 spiking rate, and so forth. What you can do is mask what would otherwise be a perfectly visible inhibition in the circuit, right? And so what we want to do is come up with kind of a new way of thinking about how to detect inhibition from null direction motion. So the idea is what we should do is specifically ask whether null direction motion is lowering the activity of the neuron, but we need to push the neuron first into sort of a range of activity where we can see that suppression, right? So what we want to do is present preferred direction motion, which causes, for instance, the neuron to become very active. Then we can present null direction motion, which in this case doesn't cause any deflection downwards. But then we can present them both at the same time. So now we're asking, how does the null direction motion added to the preferred direction motion change the activity of this neuron? And so here, this would be an opponent neuron here, because the activity is now kind of in between the preferred direction response and the null direction response. Right? Are there any questions about this? Because this is basically one of the central points. If we're at a loss here, we should clarify. Yeah.
Can you talk about ways that might go wrong? If you realize that it's not simply two inputs added together.
Yeah, so I think what we would believe is that this is a more general definition, and that it doesn't have to be thought about in this sort of model of having two opposing inputs that are already direction selective. And so one of the things we'll get to near the end of the talk will be to propose models that have these sorts of properties but don't have direction selective inputs themselves. Okay, and then just to be sure, like this definition, our definition of opponency is now when you add null direction motion, we suppress the response to an existing preferred direction response, right? And it also applies to sort of the classical direction opponent system. So here would be sort of a more classical opponent response where we have a deflection in one direction in the preferred direction stimuli. We have a decrease in activity for null direction, but when we add the null direction to the preferred direction stimulus, we would get something that would be in between. So these aren't sort of conflicting. In fact, this other definition that you're suppressing activity when you add in null direction motion is a more general view of direction opponency. Okay, so the sort of stimuli we're going to use are the sort of simplest ones you can think of in visual neuroscience. So we're going to take sine wave gratings, and we're going to move them either in one direction or the other direction. And then what we can do is we can create stimuli that are now the sum of the two, right? So now this stimulus would have a grating moving in one direction superimposed with a grating moving the other direction. So this is a little hard to visualize, so I want to show you an actual movie of what this would look like. So first, we would have a sine wave intensity grating moving to the, sorry, that went a little bit too fast. This is moving to the right. And then we'll swap in a sine wave intensity grating moving to the left here. And then what we're going to do is superimpose the two. And when we superimpose the two, you can see this sort of beating, it's called a counterphase grating, right? And this doesn't look like motion anymore, right? But this is because your visual system is already doing an opponent computation, which is zeroing out the two motion components in this sort of stimulus, right? So this stimulus contains motion to the left and motion to the right, but it's this sort of beating counterphase grating. So we're going to show this to the fly, and we want to see how different neurons in the visual system respond to this. So I want to give you a little bit of background on the fly visual system as we dive down into it. So this was our initial diagram. So this is an EM image of a fly eye, and it's covered with around 800 facets. And if you're a cartoonist, you might believe that these facets are each generating an image, and that this would be sort of a typical scene that a fly would see. And in fact, this is not true. This is not the last thing a fly ever sees. The last thing a fly ever sees looks something a lot more like this, right? So each of those facets acts as a pixel in the fly's visual system, and so there are around 800 pixels in one eye in the fly, right? And so this is a number of pixels that's actually substantially smaller than the number of pixels in an icon on your iPhone, right? So this is a very small number of pixels to use to view what's going on in the world. So sort of the non-DS processing happens in this pixel-wise format, and then the direction-selective processing also happens in a pixel-wise way. So we can move down into these earliest direction-selective neurons. Here, this is this hexagonally close-packed lattice of pixels, and in each pixel, there are 8 neurons that are detecting direction. So these are there are 2 that detect rightward motion, 2 leftward, 2 upward, and 2 downward, and then these just tile visual space, right? So this is kind of the earliest set of direction selective neurons in the fly brain, and this is what we're going to be focusing most of our attention on today. There are two types here. There's one called T4 and one called T5. We're not going to go into much detail about the distinction between them, but we're going to focus primarily on the ones in the horizontal plane. So that's these ones. So they're ones that detect front to back motion and ones that detect back to front motion, right? So that's front to back would be like this, back to front would be like that. At one step deeper, we get to these second order direction selective neurons, and these integrate information from T4 and T5 with opposite signs. So this was kind of the schematic that I showed you earlier, right? So they get excitatory input from all of the rightward tuned neurons and inhibitory input from the leftward ones, and then they integrate over really wide visual space, right? So this is what we're going to deal with, and we'll measure from HS first. So it turns out EFIS is the workhorse in neuroscience, and it's very difficult in Drosophila neurons generally. It's difficult in visual neurons in particular, and so we're going to do imaging to try to understand what's going on in the responses of these neurons. So we can set up a 2-photon microscope. We can secure a fly in place in a little hole in a piece of foil. And then we can present the fly with visual stimuli. And the really nice thing about the visual system is that the field of view of a 2-photon microscope pretty much encompasses the entire visual system. So you can see the entire visual system kind of laid out in front of you. So I'm going to show you a quick movie here of a structure called the lobula plate, which is where T4 and T5 reside. And what's nice about this structure is that it has 4 layers, and the activity of these neurons in these 4 layers corresponds to the neurons that project to these 4 layers are selectively activated by motion in the 4 cardinal directions. So what does this actually look like? This would be a typical movie. So 2-photon microscopy, remember, is photon starved, so it's very noisy. So here I've outlined the 4 layers, and then there's motion to the left, and you can see activation in this layer. And now when there's motion to the right, there's activation in this layer. And if I let the movie continue, we'd see activation in the following 2 layers. So now the goal is now to present visual stimuli, use this sort of method to measure activity in HS and T4 and T5, and try to understand the computations that these neurons are invoking in order to get their responses. So the first thing we did was we measured from HS, which is a cell that's been really well characterized historically in big flies and in Drosophila, and it's known to be the second order direction selective cell that shows classical opponency. So what we did is we expressed ArcLight. This is a membrane-bound engineered protein that fluoresces more brightly when the neuron hyperpolarizes. And so what we can do is now plot out the fluorescence of this protein over time while we present our 3 different stimuli, right? So when we present preferred direction motion, the neuron depolarizes. When we present null direction motion, the neuron hyperpolarizes. And when we present the sum of the 2, the neuron has an intermediate state, right? So this is just to confirm that our neurons do what other people have measured in the past. We can quantify this just by taking the average over the presentation, and we see that we get depolarization by preferred, hyperpolarization by null, and then basically no net response to the presentation of both at the same time. So instead of using ArcLight, we can now take advantage. So these, I don't know if you noticed, these are very small. These are like 1-2% changes in fluorescence, so that's a pain in the butt to deal with. In general, we want to use these higher, like these better fluorophores. So for instance, we can use GCamp and present the same stimuli. And so what we see with GCamp is when we present motion in the preferred direction, the calcium activity rises up and then plateaus. When we present motion in the null direction, we don't see anything, right? And so this is sort of what's symptomatic. There's now this floor effect happening either with calcium or with our indicator, right? It's not totally clear where it's coming from, but there's a floor effect such that we can't see lower activity than baseline with this calcium indicator. But when we add the null direction stimulus to the preferred direction, we get this intermediate response, which indicates that what's happening is the null direction motion is actually inhibiting or suppressing the response in HS. Okay, so again, we can quantify this just by taking the average response. So far so good. We've matched things in the field, and we've shown that this protocol that we have with the preferred direction and null direction being added on top of each other gives us a reasonable looking result there. Yes?
The impression of transient response?
Yeah.
Real or not?
I think it's real, but I don't think we, yeah, so this? Yeah, so I think there's something real there. I think the transients in this system, I think, are poorly understood generally. Okay, so what about first order DS neurons, T4 and T5? So if you subscribe to this model that I showed earlier where the opponency is generated by the subtraction of two oppositely tuned upstream neurons, first order neurons should not be able to be opponent, right? They're, like, by definition, they are the earliest direction selective cells, and they are not getting direction selective inputs. So we presented these same stimuli, and this is what we found. So here I'm showing you two T4 cell types. So this is the rightward selective ones and leftward selective ones. And in both cases, we see a strong activation by preferred direction stimulus, a minor one by null direction, and then an intermediate one when we add in the null direction one. Right? So what this says is that these neurons really look opponent in the same way that HS looked opponent. I think it's important to note that even though these traces look qualitatively similar to HS, and we know that HS hyperpolarizes to null direction motion, it's not necessarily the case that that's true with these neurons as well. So we can quantify this out here. So we see strong preferred direction activation, then this intermediate or this strong suppression here, where the preferred direction response gets pulled down by the addition of null direction motion. This is also true in T5, so it looks like T4 and T5 both have this property. So this was surprising to us, and we wanted to do a few more experiments to investigate this further. So one of the experiments we did is we added preferred direction, to the preferred direction motion, we added orthogonal direction motion, right? So this is now, there's a component sine wave moving this way, and then there's another grading that's imposed on top of it that's moving upwards, right? So the composite stimulus has upward motion and rightward motion in it. And when we did this, we saw that preferred direction plus ND suppresses, so this is the same thing I showed you earlier. And when you add the orthogonal direction stimulus, it actually looks pretty much like the preferred direction alone. And the ND plus OD also looks pretty much like the ND alone, maybe a little bit larger. And so what this tells us is that this suppression that we're seeing is actually direction selective suppression, right? So when you add in a second sine wave grading, if it's moving in the null direction, it suppresses. If it's moving orthogonal to the preferred null axis, then it's not suppressing at all. And one of the nice things about this is that the stimulus, these composite stimuli have higher contrast than the two component stimuli. In other words, the lights are whiter and the darks are blacker in these composite stimuli. And you could imagine that this was causing some kind of suppression, but here, this stimulus and the PD plus ND have exactly the same contrast distribution, and yet only one of them is showing this suppression. So we think this is a strong piece of evidence that the suppression really is a direction selective suppression of the response. And then again, this is true in T5. I'm going to keep saying that over and over during the talk, because T4 and T5 basically look the same in these assays. So next we wanted to ask what the tuning looked like for this suppression. So we could present stimuli in the preferred direction, where what we're going to do is basically keep the preferred direction speed the same. And now when we present the null direction speed, we're going to change it, right? So it can be matched, which is what I showed you earlier, or it can be twice as fast, or 4 times as fast, or half as fast, or a quarter as fast. So the first thing we did was we just took a tuning curve of what a T4 response to a single grading was moving at different speeds. So here we see the peak is at around 1 cycle per second. So that means you have like one period of this thing passing a single point each second. Then it drops off at lower frequencies and at higher frequencies. Now what we did is we picked this one, so this 1 hertz response, and we presented that consistently while we varied the speed of the null direction stimulus. And what we found was the following. So here I've just replicated this point over here. And when we added null direction stimuli with different speeds, we found this kind of nice tuning curve here that showed kind of some substantial suppression when these were matched, but even more suppression when this gets up to be a little bit faster than the preferred direction motion. And then we can take the difference between with and without the null direction and get this sort of degree of suppression curve. That's just the difference between these two curves. And then the same thing is true with T5. So we wanted to understand like what mechanisms could be involved here. And in particular, there's a sort of motif that exists in retina and in vertebrate cortex in which you have neurons with opposing direction selectivities that mutually inhibit each other. Right, so this has been found in like ferret cortex, and this is true of starburst amacrine cells in mouse retina, that when you have a population with one tuning, they inhibit a population with the opposite directional tuning. And we just wanted to see, like this could explain some of what we were seeing, and we wanted to see whether that was what was going on. So what we can do is, instead of, or in addition to expressing our fluorescent indicator, we can express a toxin that shuts down neural transmission, and that kills off these, like basically the outputs, the synaptic outputs of T4 and T5, right? So they're no longer transmitting synaptically, and we can verify this is true behaviorally, show the fly is now blind, and so forth. So if there's any kind of indirect mutual inhibition between these classes of neurons, we would presumably knock it out with this manipulation. So when we did this manipulation, what we found is that we retained this null direction suppression, right? So this feels like it is not due to this kind of mutual inhibition. And I think what's there are a couple caveats here, right? So one is like these could actually, there could be an unknown population of direction selective cells that are providing direct inhibition to these neurons. I think we have no evidence to believe that there's an additional set of first order direction selective neurons in this part of the brain. The other thing is that this shuts down synaptic transmission but not electrical transmission. So if there was some gap junction between these that led to an inhibitory circuit, we would not have effectively silenced that. And then this is also true in T5. So now we really want to think about how does this property influence our models for direction selectivity. And direction selectivity is it's a little bit difficult to think about because it's so effortless for us as humans. So when I show you this movie where I've added a lot of noise, it's just trivially easy to see that this thing is moving to the right. And that's because your visual cortex is doing a complex set of operations on this visual input in order to extract the directional information. So if instead of allowing your visual cortex to do this, I force your mathematical cortex to do this computation, what I can do is, instead of giving you intensities, I can give you just numbers for what the contrast is at each point in space over time. And it becomes very difficult. I mean, so clearly what this represents is what we want to know, right? We want to know how you would take a set of equations and apply them to these numbers in order to extract the same information that your visual cortex was doing earlier. So how should one think about this? The first thing we're going to do is start talking about space-time representations of motion. So you can think of this apple here. It's moving to the right. So at one time, it's here, and then at a later time, it fills up this box. And what we can do is sort of take each of these frames in the movie, stack them one on top of another, and then cut a slice through it this way, right? And so what we're seeing now here is a space-time representation of that apple moving through space, right? So we have time represented on the y-axis here and space on the x-axis. And we're going to ignore y. So once you've done this, once you've started representing motion in this space-time way, then direction detection is just orientation detection, right? These are exactly the same computation. And so what we could have here is we can have, for instance, this would be a sine wave grating that would be moving to the right. So this would be distant past, more recent, more recent, more recent. And so it's moving to the right. And then this would be a sine wave grating moving the opposite direction, right? And if you want to tell the difference between them, you just need to be able to distinguish this orientation in space-time from that orientation in space-time. There's this incredibly influential model for how this might work called the motion energy model. And what it supposes is that you can design a space-time filter that is matched essentially to one of these two orientations, right? So this one, you can see it sort of has this orientation in the filter, and then it's going to weight inputs positively or negatively, represented by the red or the blue. And then when we superimpose this on these two different stimuli, here you can see the red and blue lobes kind of line up with the orientation of the sine wave grating in space and time. And when it's in the opposite direction, the lobes of the grating cancel out, because what's happening is there are multiple lobes in a single positive weighting, right? And so these things are canceling. So this is essentially a matched filter then that would detect this rightward stimulus and not detect the leftward stimulus. So when we run this sort of model over our stimulus set, we get sine wave responses out. And you might think we're done at this point, right? Because it's very clear that the preferred direction stimulus generates this high amplitude sine wave, and this null direction stimulus generates this low amplitude sine wave, right? And what we're going to do, though, is we're going to require that in order to be direction selective, you have to have a mean response that's different in the preferred direction from the null direction, right? And this is not some arbitrary definition that we're using. This is actually what you have to do, because if what you said is that, Damon, that's crazy, I can just tell that this is a direction selective signal, right? The amplitude here is high and the amplitude here is low. But what you're doing when you're saying the amplitude is high and low is you are taking an implicit nonlinear transformation of this sine wave, right? So in order to calculate the amplitude of a sine wave, you have to perform a nonlinear operation yourself, right? So you can't get away from taking the mean. And since we want to know the full computation, we want to know what nonlinear transformation you're taking on the stimulus. So maybe the simplest one could come up with is just to threshold rectify these signals at 0, right? So we'll do that. And now all of a sudden we can take the mean and we'll get two different answers, right? So before the mean of all of those sine waves was 0. Now the mean of the high amplitude sine waves is larger than the mean of the low amplitude sine waves. And we can now get a direction selective signal that has a large response in the preferred direction, a small response in the null direction. So this is the motion energy model. And these, I think it's hard to overstate how widely these have been applied in motion computation, and it's a form of LN model which has been widely applied throughout neuroscience. But in motion computation in particular, this has been applied throughout the primate literature. Recently, it's been proposed to explain direction selectivity in Starbuck-Amakrin cells in mouse retina. It's also recently been proposed to explain direction selectivity in mouse cortex. Also recently, it's been proposed that this could be an option to explain direction selectivity in T4 and T5. And this is based on a set of beautiful measurements of membrane voltage in T4 and T5. And this is an example from one of the papers that did this. So they recorded electrically from T4 and T5 and measured a receptive field and showed that the membrane voltage in these cells was largely looked like a linear transformation of the stimulus. And so this has kind of led, like this fact that the membrane voltage is a linear function of the stimulus has led to, I think, both explicit and implicit models in the field for how direction selectivity could arise in T4 and T5. And that model basically looks like this. You take your stimulus, you filter it with your linear space-time filter that has some orientation to it. You get out your membrane voltage, which is now a linear function of the stimulus. And now we have a transformation to calcium, which is nonlinear. And that gives us a calcium signal. And now this calcium signal has a mean that's direction selective, right? And this would be consistent with all of the data in the field. And this sort of transformation for voltage to calcium is a very standard-looking curve that people would sort of widely believe is reasonable. So what we actually did in lab, and a talented undergraduate lab proved, was that if you take an LN model with some kind of convex nonlinearity, right, so that these would all be examples, it cannot generate a direction opponent signal, right? So there's no way that this sort of model that has any space-time filter, right, so any linear filter preceding a convex nonlinearity cannot generate direction opponency. And so what you get out is you always get that when you add the null direction motion, you always get out a signal that's equal to or larger than the preferred direction response, right? And so what this says is, the converse of this is that if you measure direction opponency in a cell, then this sort of LN model cannot explain it, right? And so it's kind of rare that you get to rule out a class of models kind of with such generality. So I decided to put a big X on this. But I think the confidence that we have in this is primarily, so the mathematical proof is just there. I think the lack of confidence would come from things like, did our silencing experiment work as well as we thought it did? Are there electrical junctions and that kind of thing, which would still not permit an LN model to explain the response, but would allow there to be sort of other options. So we wanted to look into what kind of models could actually account for this sort of property that we measured in T4 and T5 and the direction selectivity. So in LN models, the nonlinearity is static and doesn't change in time. And so one thing you can do is mess around with the type of nonlinearity and make the nonlinearity itself a function of the stimulus. So this could look something like this. So here we have our oriented space-time filter, which is being acted on by this nonlinearity in order to get the direction selective signal. And what you can do is have a second linear filter, which now is not the, that doesn't get acted on by this nonlinearity, but acts on this nonlinearity and, for instance, adjusts the gain on the nonlinearity. And if you set this up correctly, you can generate, and notice this is not direction selective, right? So we have a non-direction selective gain adjustment, and then an oriented space-time filter coming in, and these two linear filters of the stimulus interacting via gain changes in this nonlinearity. So you can design or construct models like this that then produce the required suppression that we see when we add in the null direction motion. The kind of like nicer version of this, I think, is to actually just go back to basics and what we know about the anatomy and the function of the fly visual system. So there's, I guess, in the fly visual system now, we have a complete connectome from photoreceptors all the way through to T4. And so we actually know what all of the inputs to T4 are, where they look in space, and then functional studies have actually measured their properties. So this is sort of a summary of what is believed to be true about T4 function and anatomy. So it gets a central input via MI1 and TM3. And these are on cells, meaning they depolarize to light increments. And then it has two flanking inputs that are very likely to both be inhibitory. One of which comes from an on cell that has pretty slow dynamics, and one of which comes from an off cell, which has pretty slow dynamics. And this is sort of like why this off cell is in here. I think no one is particularly sure. And so one of the things I think we can explain a little bit about is like why this structure might exist in the fly brain. So what we can do is build a sort of very simple computational model that's based just on the anatomy and functional properties of these effectively 3 neurons that are upstream of T4. And so what this looks like is the following. So we have 3 inputs at 3 different points in space. And what we're going to do is sort of delay the 2 flanking ones and then model synaptic inputs to T4 and measure the membrane voltage in T4, and then have a static nonlinearity that acts as the calcium transformation at the end of all this. And when we do this, we find that ND motion actually pretty strongly suppresses the response in this model. So I haven't told you why, right? Like I've just told you that this is a fact about this model. One thing that's clear is that in order for this model to work, these cannot just be sort of current inputs to the membrane voltage, because then this would just be an LN model, and we already proved that an LN model can't possibly account for this type of suppression. So we wanted to think a little bit more about how the structure of these inputs was causing this response. And the important thing we did is we modeled these inputs via the postsynaptic conductance into T4. And what this means, because we're modeling conductance rather than current injection, as the conductance gets larger and larger, you get sort of a saturating current that goes in, and conductances can interact with each other, right? So one conductance can shunt current from another conductance. And it turns out this is essential to why this model works. So what we can do is neglect capacitive currents and then just write down an expression for what the membrane voltage looks like as a function of these conductances. And this is very typical for what you would get. You have a conductance-weighted reversal potentials up in the numerator, and then the sum of all of the conductances in the denominator. And this is something from first year neurophysiology course. And what's nice, though, is that each of these conductances here is closely related to space-time filter of the stimulus, right? So what we can do is actually expand this numerator to find out what an effective receptive field is for the numerator. And we can also expand this denominator and find out what an effective receptive field is for the denominator. And what we find is that in this case, the numerator has an effective receptive field that has this orientation in space-time. And the denominator has an orientation in space-time that's exactly oppositely tuned, right? So it has the opposite directional tuning. So what this means is that when you have motion in the preferred direction, you're activating the numerator. And when there's motion in the null direction, you're preferentially activating the denominator, right? Which is suppressing the whole response, right? So this is, instead of being this sort of subtractive mechanism that was seen in like second order cells where you're subtracting 2 oppositely tuned directional neurons, here this is actually a suppressive, divisive suppressive mechanism, right? So it's essentially changing the gain of the neuron to lower the gain when there's null direction motion. Okay, so I want to finish up by asking sort of why these first order direction selective cells might want to have these sorts of opponent properties. And I think it's clear, so that there have been a lot of studies that have posited why second order direction selective neurons might want to be opponent. And they're mostly based on wanting to be selective for specific visual flows, right? And so if you want to be selective, for instance, to all motion moving in one direction, or motion in one direction at all points in space, then you want to not respond, for instance, to half of the field moving one way and half moving the other way, and this subtractive suppression does that exactly. But those sorts of things can't really apply to like a local motion detector, right? Like this is the atomic motion detector in the Fly visual system. There's no sort of smaller field of view that can be cared about. And it presumably wants to get out like what the net motion is inside its receptive field. So the thing we've been thinking about is essentially that there's noise in these motion estimates and noise in these motion signals, and the noise is often non-direction selective. So when you run a motion detector over a natural scene, then it has a direction selective signal, but superimposed on that, there's actually a non-direction selective noise term. So you can think about, for instance, moving this forest scene in front of two detectors with opposite directional tuning, and the responses you might get could look something like this, where there's definitely a difference. So the one with the preferred direction in the direction of the stimulus has a higher average response, but there's this fairly large non-direction selective noise, right? So this is noise that's independent of the direction of stimulus motion that's superimposed on top of it. If what you did is instead of computing these two, you computed the difference between these somehow, then what you would get is a response that is much less noisy, right? So the standard deviation relative to the mean here is much smaller than these guys. So essentially, if you could do this kind of opponency early on, you might think that you could devote more of your dynamic range as a neuron to transmitting information about direction rather than this non-directional noise that might come in natural scenes. So to look at this more carefully, we did some simulations where we took natural scenes and then we slid them in front of different opponent and non-opponent direction selective models at different speeds. So we had a distribution of speeds, we selected scenes at random, and then we just got an entire distribution of stimuli and responses, right? And we want to ask, how much can we know about the stimulus velocity given a response, right? And so we have this non-opponent model. So this is just sort of one of the simplest direction selective models in the literature. And then we have an opponent version of it, where it's this model, and then we subtract the spatially symmetric signal here. And one thing we wanted to do is we didn't want, like, so when you have an opponent model, all of a sudden you can have all these negative responses. We didn't want just this increase in dynamic range to be responsible for any improvement. So we stuck this rectifier on the end. So now both of these models give all positive responses, and we just want to know, like, how different are they? And in particular, if you know the response, how much do you know about the stimulus? And so this is now the distribution of velocities conditioned on the response. So our response is arbitrary units here, and we show it going from 0 to 6. And what you can see is that there's this very wide distribution of velocities that's sort of independent of the response, right? So this actually is not a very good at telling you what the true velocity was in your stimulus. If you look carefully, you can get a little bit of information out of it, so it's not nothing, but it's not very good. If what you do instead is you do this opponent subtraction, now all of a sudden, if you see a positive response, you can, with quite high certainty, know that you had a positive velocity, and if you see a 0 response, you can know with some certainty that you had a negative velocity, right? And so what's happening here is this noise, like the fact that this isn't working well, is generated by structure in these natural scenes, which is generating responses or noise in the response that has nothing to do with the stimulus velocity, right? So that's fundamentally what's happening here. And you can quantify this with a generalized correlation, which roughly, it tells you how much variance in the velocity you can account for by knowing the average velocity given a response. So this is just sort of a non-parametric correlation coefficient. So clearly, how much improvement you see here depends in large part on your algorithm for your direction selective response, right? So some algorithms are going to be more sensitive to like the structure of natural scenes, and some of them are going to be less sensitive to the structure of natural scenes. But over several models and cases here, we always see that natural scenes have this very noisy sort of response in standard motion detection models. And when you subtract oppositely tuned versions of this, you can massively improve your motion signal. Okay, so I think I'll stop there. So what we found is we found that direction opponency in flies emerges at the first direction selective cells. And so this is sort of different from where opponency has been seen in the past in many direction selective circuits, where it's primarily been observed in second order neurons. And the mechanism that's been provided in those cases has been that you have two inputs with opposed tuning, one of which is excitatory and one of which is inhibitory. And so it's sort of surprising that we see this same property in first order neurons. And I think importantly, this result seems to exclude a broad class of very popular models for direction selectivity. And what this means, I believe, is that we're going to have to look more carefully at what the actual nonlinearity is, and static nonlinearities are going to be unlikely to cut it with this system. And then last, we showed with simulations and natural scene statistics that this type of opponency that we observe in these first order cells can actually improve direction selective signals in a substantial way. And so it's actually maybe not that surprising that you would find it in the earliest direction selective cells, because this improvement should improve signals wherever, right? And there's no reason to put up with a bad signal at any point in your visual system if you can make a good one. Okay, so I want to thank my lab and funding, and in particular, the work I talked about today came from Bara Badwan and Matt Creamer, who are graduate students in the lab, and Jacob Zavatone-Beth, who's a talented undergrad in lab. And so with that, I'm happy to take questions. Thank you. Yvette.
Really nice talk. Let me just make sure I understand the last section. So you're comparing these kind of basically a half correlator with a full correlator.
Yeah.
And my understanding from memory of working on this is that one of those is going to be velocity tuned, whereas one of those is going to be temporal frequency tuned. And I was wondering if you could talk about a little more. Is it just that being temporal frequency tuned helps you because that well matches the spatial correlations in a typical natural scene?
So I think, so it's true that depending on what your filters are, you can make that the subtractive version of this is always temporal frequency tuned, and that the individual multiplier version can be velocity tuned. I'm not sure that specifically relates to the result. I'm not sure that property of these is what relates, or is what generates the results that we see here. I think the primary thing that's generating the results that we see here is that, roughly this is common mode subtraction, I think would be the way to think about this, right? So common mode subtraction in an amplifier, you want to know what the difference is between two inputs, and when the reason the single multiplier model is responding with all this noisy input is because there are spatial correlations in the input. You get bright inputs to both the delay line and the non-delay line, and then those are multiplied together, and that's direction non-selective, right? So it doesn't matter what direction it's moving in, those are going to get multiplied together and add to the signal kind of no matter what. And those are the sorts of things that are being subtracted off when you take an equivalent mirror symmetric version of this and subtract it. Does that make sense? So I think it is. I wouldn't. It's at least not superficially related to this tuning difference between those two models. Yeah.
So I assume that the noise that you observe in the visual scenes, for different parts of the natural scene, the noise will not be correlated. Is that correct? So if in the end anyway what you're interested in is does the
Yeah, so I think this is a good question, right? So do you really need to do this? Because HS, for instance, downstream neurons are just averaging over visual space anyway. And so maybe you can just average away that noise spatially. Am I paraphrasing the question right? Okay, so I think, so this is definitely a possibility. It's the noise is not uncorrelated between, so there's sort of a correlation length that depends on like the correlations in the scene, right? So like faraway points will be less correlated than nearby points, but you won't get perfect independence anywhere. There's also this question of like correlations over scenes, right? So different scenes themselves have different levels of contrast and that kind of thing, and you want to be able to, you want downstream processing to work regardless of that as well. I think, so I guess so the take home there is averaging doesn't work as well as it might if these were independent, right? But it still works. I think there's one, so there are a couple cells that seem to take T4 and T5 input and have much smaller regions that they pool from. So there's a loom sensitive cell that looks like it takes input from only a small fraction of the visual field. And so you could imagine there you get to average less and you might want this more. I think even if you're averaging, like it's still good to use so it's good for the T4, T5 cells to use most of their dynamic range to signal direction and not some other aspect of the scene that downstream neurons might not care about. So I think it may be helpful for these sort of more local downstream computations, but I think it's also very helpful even if you're just straight up averaging these things later on. Yeah, Kenny.
Confused about whether I should be thinking of these as operations underneath a pixel, operations on a pixel, or something more about the scene or the whole field, and in particular, if I should be thinking about self-motion, where there seem to be other kinds of flow fields that aren't.
Yeah, so I think the motion signals that we're thinking about are on they're computed at each pixel in space, but they use information from several local pixels. So it's roughly a group of, let's say, 7 pixels, like a center one, and then the 6 surrounding ones. And so that's a good cartoon of what T4 at least is known to do or is thought to do anatomically based on EM reconstructions. So that's the first question. The second question is about flow fields, right? And I think this is a really important one because what we did is we rigidly translated these scenes, which is the equivalent of what the motion is you would see if you were just rotating without translating in the world. And I would say largely we chose to do that because it's possible and we don't have good data sets with 3-dimensional information about translation through the world. So we have a lot of trouble doing that. I think it's also very clear that T4 and T5 feed into circuits that regulate the rotation of the fly specifically. So it's what's possible, and it's also what's largely appropriate. They also feed into other circuits that regulate translation and other things. And there, I think we don't know as much about how they would respond to those sort of more interesting flow fields.
You're potentially missing other axes of rotation and
So I think other axes of rotation like all do the same thing where they rigidly translate the world, and there's not like relative motion of different parts of the world. So any rotation of your body, like there aren't sort of objects that are occluding one another as you turn and that kind of thing. So what I think we're really missing is this translation. And then you could, like I think we only study these horizontally tuned T4 and T5, but like I think from EM it's pretty clear, at least with T4, that the vertically oriented ones look anatomically similar. Okay, great, thank you. Oh, Alan.
Yeah, does the model you showed at the end with the role of opponency on natural scenes, does that hold if you do divisive opponency, like this conductance model suggests?
So I think what's really hard is to figure out what your apples to apples comparison should be for this type of model. Because I do not know what the apples to apples comparison is for that divisive one. I guess like one thing I think we didn't think of at the time, but now that you ask, we potentially could do is have like we could just take the numerator here and not have the denominator. I'm not sure whether that really is, and so that would be the non-opponent version, and then you stick in the denominator, and then you have an opponent version. I'm not really sure that's apples to apples. Like our goal was to figure out, here we've got like identical tuning, all the delays are the same, all the inputs are the same, all the operations are the same, and all we're doing is subtracting a mirror symmetric version of the signal. So I don't know the answer. And I think I don't know it because I don't know what I would test to say x is larger than y in order to answer that question in a sort of nice controlled way. Yeah.
Do you know that you need three terms? Like I think you might see why the third.
So I think you do not need all three, but I think this is actually the cutest thing here, is so these inhibitory reversal potentials. These inhibitory reversal potentials here are negative relative to 0. The excitatory one is positive, and then G1 is an off cell, or G1 is off, and G3 is on. And what happens here is you've got like these three lobes. So you can imagine this is cell 3, cell 1, cell 2. And then what happens between the numerator and the denominator is the two lobes just switch sign, right? And it's because this inhibitory reversal potential has a negative sign associated with it. And that, with this setup, it gets you to flip the directionality of the input. I think it's very likely that it would still work like without this, right? Because those are still tuned. So I think we don't know why it's 3, but I think we have a good we have a nice explanation for how this off cell could be contributing in an interesting way in terms of making these oriented receptive fields. Okay, terrific. Thank you, guys.